Hello everybody, this is my science video tutorial on anaerobic respiration, alcoholic fermentation, lactate fermentation, and whatever the heck else I can think of to throw in there. This is my worksheet here that you can see in front of you for alcoholic and lactate fermentation, and it's available on my website, www.zerobio.com, and I'll put a link to that in the information section of this video, and also a direct link to the worksheet. And my suggestion would be, if you like to, download the worksheet and follow along with the video, and you get the best of both worlds, if that makes sense. Okay, so we're going to talk about alcoholic fermentation, but just before we do that, let's just look at a little overview here and see if we're all on the same page. So if we start up here with glycolysis, right? Glycolysis, the breaking down of glucose into pyruvate, making ATP, NADH, that kind of thing. As you know, glycolysis leads into matrix reactions. I'm going to summarize that and just call it Krebs cycle for the moment here. All right, because that's the main part. The other part is pyruvate oxidation. It's one main reaction. After that, you're going to get the electron transport chain or electron transport system or oxidative phosphorylation. All right, so these are the three steps of aerobic cellular respiration. And that really implies that we need oxygen. And oxygen, of course, is only required for these parts right here. The first section, glycolysis, is considered anaerobic, doesn't require oxygen. The reason that we need oxygen is, as you may know, oxygen is the final electron acceptor in the electron transport system. And it allows the electron carriers, NADH and FADH2, that have been produced in glycolysis and in Krebs cycle. It allows these electron carriers to drop off electrons at the ETS, and that's its role, the electron transport chain or system, to transport those electrons. And as those electrons move, they create the ATP, but it also causes the oxidation of those electron carriers back to their original forms so that they can go back into Krebs cycle or back into glycolysis for reuse. So those electron carriers are reoxidized, continuing the metabolic pathway to create ATP. So as long as oxygen is available, things are aerobic and everything works out. But what if oxygen is not available? As you may know, glycolysis is anaerobic, doesn't require oxygen, so put minus O2 as opposed to plus O2, needing oxygen, or with oxygen, without oxygen. But there still is the production of NADH in glycolysis, and that requires an input of NAD+, the oxidized form, to make this. And as long as that is available, then ADP will be converted to ATP. I'm not worrying about how many right now. But, you know, if you only had one NAD+, and you converted it to NADH, and that allowed you to make ATP, that would be it. You'd do it one time, and the whole thing would stop. You'd live for about 1 second, and you'd have no more ATP, because you need this to go back to this. Without these steps in oxygen, how are we going to do this to keep glycolysis going? And that's the purpose of fermentation, to keep glycolysis going so that we can, keep creating ATP. And in order to keep doing that, we need more NAD+, but we're using it up every time we do glycolysis. Enter fermentation. So let's have a look at the fermentation reactions. So on this worksheet that I made for you, we've got glucose up here, 6-carbon molecule. I haven't drawn all the atoms on there, just the skeleton so that you could see it. You could also color this in, right? You could color the carbon black, the oxygen red, and the hydrogen white or something like that if you wanted to make this colorful. All right, and then you could follow the carbon atoms all the way through. All right, alcoholic fermentation. This is a process that is occurring in things like yeast, single-celled microorganism, anaerobic bacteria. This is not something that is occurring in humans. We do not do this process. All right, step 1, we're going to begin with glucose. So write this in on your worksheet. Glucose, one glucose molecule with its 6 carbons, will undergo glycolysis. Whether oxygen is present or not, it's still going to occur. So this arrow represents the biochemical or metabolic pathway of all of glycolysis and simplifies it from glucose all the way down to the production of pyruvate. This is happening in the cytoplasm of our cells or in the yeast or in the bacteria. 2 pyruvates are made per glucose. Notice there are 3 carbons in one pyruvate, 2 of them produced. That's our 6 carbons. All right. Along the way, NAD+, 2 of them are converted, that's what this arrow implies, by interacting with glycolysis, to 2 NADHs. And these little circles might represent the 2 electrons that are being carried on each NADH. All right, so write those in. That is the reduction of NAD+ to NADH. Now, glycolysis takes 2 ADP molecules and produces 2 ATP. That's net. And you can see I've got 2 phosphates on here, so this is adenosine diphosphate, and 3 here for adenosine triphosphate. So we produce 2 ATP net, and we produce our 2 NADHs, and we produce our 2 pyruvates. So we can come over to the summary over here and write in glycolysis produces a net of 2 ATP, 2 NADH, and 2 pyruvate. And that's our typical glycolysis summary that if you've learned glycolysis before, you'll be familiar with that. All right, great. So we've done it once. We made a couple of NADHs and some ATP. Well, without oxygen, nobody's going anywhere. We're going to live for about 1 second, and that's it. Fermentation is the process of converting pyruvate made by glycolysis into a final product here. All right, and there's 2 types of fermentation. This one's going to be alcoholic, so we're going to make alcohol. But first, let's go back here. Pyruvate has a carbon dioxide removed from it. So write carbon dioxide down here. Now there are 2 pyruvates, so each loses a CO2. And if you keep track of the carbons, 3 carbons in pyruvate minus 1 carbon in carbon dioxide leaves a 2 carbon molecule. The removal of CO2, which is essentially a carboxyl group, is called decarboxylation. The decarboxylation of pyruvate leads to this product, which is an aldehyde. See the C double bond O and the H. This is called acetaldehyde. So the first step of alcoholic fermentation after glycolysis is the production of acetaldehyde by decarboxylating pyruvate. Then this molecule, acetaldehyde, is reduced by NADH. NADH was produced in glycolysis, and then once it's made, it gives its electrons over to acetaldehyde, and it produces alcohol, ethanol, that particular type of alcohol. Notice ethanol has 2 carbons, so does acetaldehyde. So we can conserve our carbons by looking at this. There are 2 here, times 2 is 4 carbons, and these 2 times 1 is 2, that makes 6 carbons, our original glucose. We haven't lost any carbons, we've accounted for them. But by reducing acetaldehyde, we regenerate NAD+, which can go back into glycolysis to continue to make 2 ATP. And that's why this is happening, so that ATP can be continually made to fuel the life of a simple yeast or a simple anaerobic bacterium. So let's go on in the summary. In the absence of oxygen, pyruvate loses a carbon dioxide and is converted into acetaldehyde. That's this step here. This is then reduced by NADH. The acetaldehyde is then reduced by NADH, producing ethanol, which is our alcohol, and regenerating NAD+ for reuse in glycolysis. So this is the solution to the lack of oxygen. Can't go into Krebs cycle, can't go into ETS. And you might think, well, some of these organisms don't have that anyhow, but there are some that do. And if oxygen's available, they'll do the 3-step aerobic cellular respiration. If oxygen is not available, they can do this. So for simple organisms like yeast or anaerobic bacterium, this is enough for them to survive. Alcoholic fermentation, the buildup of alcohol, a way to regenerate NAD plus to keep glycolysis going. We can't do this, all right? Otherwise, we would just hold our breath or do exercise. Alcohol would build up, and that would not be a very adaptive situation for us, all right? We all know that when we do exercise, yeah, we get sleepy or whatever, but we don't get intoxicated. So let's look at another process. Okay, I'm going to change the screen here. I'm going to go on to a different type of fermentation. Lactic acid fermentation or lactate fermentation. This is the one that happens in us. Once again, we're going to start with glucose. All right, so write glucose on this new worksheet. Same structure. Glucose will again go through glycolysis and produce our 2 pyruvate molecules, 2 3-carbon pyruvates. In the process, a couple of ADPs will be phosphorylated to ATP. This is substrate level phosphorylation, right? Phosphate coming off an intermediate in glycolysis, making ATP. NAD plus will be converted or reduced to NADH. We've made our electron carrier, our reduced electron carrier. But we're not going to produce alcohol now. So imagine this is happening in our bodies under somewhat anaerobic conditions. Now, you probably know, if you've ever been swimming and gone underwater, you can hold your breath, right? But you can't hold your breath all day. You can't do this all day. You can hold your breath for a couple of minutes, let's say, and you start to run out of oxygen. And if you don't breathe again, get oxygen, you're not going to make it very long. All right? So we cannot survive in strict anaerobic conditions. All right? But when we exercise, we're out of breath. That basically means, when we're out of breath and breathing hard, that we don't have enough oxygen. If you had plenty of oxygen, you wouldn't be breathing hard. I mean, we just think that, we do exercise and we just start to hyperventilate. But think about the physiological reason for that, right? You don't will yourself to breathe hard. It happens as a response to the low oxygen. Well, actually, maybe it happens as a response to the high CO2 or the change in pH in your blood. There's a bunch of things physiologically going on, right? As you use up glucose and you do aerobic cellular respiration, if you've got some oxygen, you start building up CO2. CO2 in high enough concentrations can be toxic. You have to breathe that out. So one of the reasons you start to hyperventilate or at least increase your breathing is to blow off the CO2. All right, that's going to change the pH of your blood and stuff like that. But if we just think of it in terms of oxygen for now, if there's not enough oxygen, you're not making enough energy. Your body knows this, causes you spontaneously or involuntarily, is probably the better word, to breathe deeper and faster, saying, get rid of the CO2, but give me some oxygen so I can make some real energy because I'm working here. My muscles are working hard. So again, what I'm trying to say is you can't survive without oxygen indefinitely, and you need a method to survive when oxygen is just being depleted to some extent. All right? So let's say during exercise. So imagine we've got these muscle cells. You got a supply of glucose there, breaks it down to pyruvate. You make your NADH and you make a little bit of ATP. All right? Not enough to survive on for any long period of time. Okay, but we're only going to exercise for a little while. Okay. Pyruvate then is the molecule in lactate fermentation that gets reduced. NADH reduces pyruvate, not acetaldehyde in alcoholic fermentation. NADH reduces pyruvate, converting it to lactate. Notice this is a 3-carbon molecule, just like pyruvate. Okay, so we haven't lost any carbons. Lactate is what builds up. Lactate, lactic acid. That regenerates NAD+, which can go back to glycolysis and keep this process going for a little while, and we'll survive. Now remember, you're not locked in an airtight container, right? If you're exercising, there's still oxygen in the air and you're breathing harder. There's just less of it, all right? So some of your tissues might be doing a little bit more of this and some still have enough oxygen. So on the whole, you're not completely anaerobic, all right? And this is a way to deal with that sort of less oxygen amount. So not every glucose molecule gets the opportunity to go all the way through metabolism. Some of them say, look, I'll do this. You go through aerobic cellular respiration. There's enough oxygen for you, but there isn't for me. So this is another way to generate some ATP and regenerate the NAD+. Okay, so let's look at the summary. Glycolysis produces 2 ATP, 2 NADH, and 2 pyruvate, just the same as before. In the absence of oxygen, the pyruvate this time is reduced by NADH, producing lactate instead of ethanol, and regenerating the NAD+ for reuse in glycolysis. Now the lactate, being an acid, lactic acid, starts to sting your muscles, and that causes some discomfort as it builds up. But if you stop exercising and repay that oxygen debt, the lack of oxygen, this stuff will go back into the glycolysis pathway, be fully metabolized, and things will go back to normal. All right, so alcoholic and lactate fermentation are methods to regenerate NAD+, to keep glycolysis going, to make a small amount of ATP, and that's why they exist. Those are the fermentations. All right, so we've got that central aerobic cellular respiration pathway, and we've got some branches coming off as fermentation reactions. Now, there's one more thing that I want to talk to you about. Doing that again. Okay, so I'm just going to do a little bit of drawing here as we summarize. We know that glucose is the major fuel of the body. Just as if you were driving your car and you pull up to the gas station, right? You're going to fill it up with gas. You're not going to go into the convenience store and buy some Pepsi and put it in the gas tank. That probably wouldn't be smart, right? Even if your car ran just kind of weirdly, it's going like, give me gasoline, okay? That's my main fuel. So that's what the body needs. It needs glucose. That glucose will then go down through Krebs cycle. It will go down through the ETS. I'm just going to be short here, all right? Now, glucose is a carbohydrate. And there are other carbohydrates that could feed in here. So imagine starch going in here, a polysaccharide of glucose, made of glucose monomers, or glycogen, or other disaccharides that could be split, and you get glucose and fructose and all that. This is the central pathway which generates a lot of the ATP that the body needs, the pathway involving carbohydrates. Now, when you eat some food, let's have a hamburger, let's say, all right? Sure, you're going to get some carbohydrates in there, but you're also going to get lipids or fats, right? You got cheese in there. There's fat in the meat of the animal that you're eating. You're going to get some proteins. You may even get some nucleic acids. Well, of course you are, because you're eating the cells of the cow. You're going to eat the cell, break open the nucleus. You're going to get the cow's DNA. You're going to use those nucleotides to build more DNA for yourself so your cells can divide. But let's not talk about the nucleic acids right now. Let's just focus on this stuff. All right. So think about the biochemistry of lipids. Let's just look at triglycerides for the moment, all right, and not deal with cholesterol and stuff like that. Triglycerides, as you know, are made from glycerol and fatty acids. Okay? Now, think of glycerol, 3-carbon molecule, all right? Is there anything in glycolysis that is a 3-carbon molecule that has a similar name and hence similar structure? What about that glyceraldehyde stuff? So when you eat your fats, they can be broken down and feed into glycolysis at the appropriate spot, at the glyceraldehyde stage. So yeah, sure, you missed this stuff up here, but who cares? You're going to see the rest of the show. You missed 5 minutes, you got the rest of it, all right, to make lots of ATP and NADH and stuff like that. So this is how your fats can enter metabolism, all right? Now think about your fatty acids, right? If I draw a fatty acid here with a carboxyl group right there, and if I just draw arbitrarily a big, long carbon chain here, I'm not going to worry about any of the other atoms. Well, there are enzymes that can split this fatty acid. And of course, in triglycerides, maybe you get 3 fatty acids. And when you eat a big chunk of fat, you don't just get 1 triglyceride, you thousands and thousands. Think of all the 2-carbon molecules you could get from a big chunk of fat. Well, where do you know a 2-carbon molecule could feed into the glycolytic pathway or the metabolic pathway? Well, what about up here in Krebs cycle? If I draw it like this, acetyl-CoA is right there. So every one of these 2-carbon molecules feeds in at the level of acetyl-CoA. This is a very short fat. If I had a really long fat that was 16 or 32 carbons long, think of how many acetyl-CoAs you're going to get. Now, once again, you're going to miss a little bit of the show. You're going to miss all this stuff. But you're going to feed in at this level with all these acetyl-CoAs. You're going to make a lot of NADH. You're even going to make some ATP there and a lot of FADH2s that are going to go into the ETS. You're going to make a lot of ATP. And that's why fats have a lot of stored energy. When you break them down, they can feed into multiple places in this metabolic pathway, creating a lot of energy. That's another reason why when you eat fatty foods, sure, you can use some of that fat for energy, although the body goes, I really want my gasoline, right? And you're going to say, well, I'm going to store that fat. And we may not like it, and it may not be overly healthy if we have too much, but we need some fat. Fat keeps the body warm. It insulates organs. It supports them. But it's also a really good energy reserve. And you know from people that say, I'm going to go on a diet, I'm going to do more exercise, I want to burn fat, that you can indeed burn that fat. Now, of course, when I say burn that fat, think about this for a second. When you burn something, switch colors here, you generally need oxygen, right, to have a fire. So if you want to burn your fats appropriately, you're going to need oxygen. What does that mean? Well, if oxygen's available, that means Krebs cycle is going to work, right? That's the aerobic part. And so is ETS going to work. So with oxygen, it means your fats can feed into these levels and continue down for the rest of the show, Krebs cycle and ETS, making a lot of ATP. And you're burning your fat. You're getting rid of the fat. You wanted to lose weight, you're burning it. If you don't have enough oxygen, all right, then all you can do is this upper part. That's not going to burn all that much fat. The most of it is going to come down here. You want to split the fatty acids and use them up here. If all you're getting is a little 3-carbon glycerol to go into glycolysis once in a while, you're not, and then no further, right? You're not going to burn much fat. So you need to do aerobic exercises with oxygen, not to be too much out of breath. So fats enter the glycolytic and Krebs cycle and ETS, those pathways. All right. Proteins, just quickly. Carboxyl group, carbon with an R group, H, amino group. Okay. You are going to chop off the amino group when you eat your hamburger and you get the meat, the fat, the meat and the protein. Chuck the amino group. We'll come back to that in a minute. This is probably going to come off as a carbon dioxide. Decarboxylate it, breathe it out, get rid of it. Think of all the carbon stuff you have in the R groups, all right? All kinds of carbon chains. They can feed in all over the place, okay? So proteins can also be used for energy, although it's not a primary source. The body doesn't really use proteins very much. Again, it prefers the carbohydrates, or it will use the fats. You use proteins to build up your own protein in your body, to build up your muscle and your antibodies and that kind of thing. All right? But you can use protein as well and get energy out of it. Now, those amino acids, though, if you cut off an amino group, this nitrogen stuff is bad stuff. This can form ammonia, which is toxic. You have to get rid of the amino groups. And so what the body does is it takes one amino group, clicks it onto a carbon with a double bond, oxygen, and another one, and this becomes a diamide. Look, there's an amide. Here's another amide. This diamide is called, let me switch colors one more time. This is called urea, which is one of the major nitrogen products in urine, right? Urea, urine. It's highly polar, all right? It's water soluble, so you urinate it out. You get it out of the body because it's toxic. This is why you have to go to the bathroom, okay? So all of these macronutrients in your diet, including the nucleic acids to some extent, can feed, I just want to scribble all over this, into the metabolic pathway, usually this central pathway at different levels to generate energy. The primary one is the glycolysis, utilizing carbohydrates. But fats can go in, proteins can go in. And by the way, one more thing, you can convert your glucose into fat. People talk about, I'm eating a sugary chocolate bar. You're eating so much sugar, you're going to get fat, right? If your body goes, you know what? I have so much carbohydrates. I don't need to make any more energy. I have so much sugar. What am I going to do with it? I think I'll save it up, right? I'll invest. You invest in fat. You start building it up. You can also create proteins, all right? That's the complicated physiology and biochemistry. Anyways, I hope that was interesting, and I hope it helps. Thank you for watching. See you again soon.